Good day, everyone, and welcome again to the second part of the two-part webinar on PanGenomes. My name is Sheetal Dixit. I'm Director of Business Development here at KeyGene, and I will be your host today. I want to thank you, first of all of you who joined our first part of the PanGenome webinar and gave your valuable input, which had really helped us. And please continue to do so. I hope that you will notice that we have tried to incorporate most of your input and the feedback for today's webinar. So once again, thank you very much. We're also very thankful and very glad that today, for today's webinar, about 360 people have registered for this webinar. And we really look forward to have another exciting hour for you today. So for today's PanGenome webinar, we will be focusing on construction of PanGenome and the applications of it. And for you all, we have fantastic speakers for you. And I would like to present our guest speaker, Professor Ellen Session, who will be joining today from the US. Hi, Ellen. Welcome.
Hello. How are you?
Very good, thank you. And two of my colleagues, Erwin Datuma and René Hofstede, are joining me live here from the KeyGene's studio. So if you have questions, you know my other colleague, Eric Toussaint, he's joining us from backstage of the studio, and he will be taking your questions. So if you have questions, please do. Hi, Eric. Please do type that in the chat box, and Eric will help us bring that to the table for our speakers. We will be taking all the questions at the end of this webinar. So please don't forget to type them in. Well, let's start the webinar, and that gives me the opportunity to introduce you to our first speaker, Dr. Erwin Datuma. Erwin is a senior scientist, genome informatics at KeyGene, and last time, Erwin introduced us to the concept of PanGenomes, and today he will be giving us an overview of the state-of-the-art technology and the tools that we have been developing here at KeyGene for PanGenome. Erwin?
Thank you, Sheetal. So, indeed, today I would like to continue the story that I started with previous month. So there my co-worker Alexander and myself, we showed how we are now at KeyGene unraveling genomes at an unprecedented scale. So for many of the crop species that we work on, we are now sequencing multiple genomes at high quality, at high contiguity. And in order to deal with these very large data sets, we have also developed a comprehensive computational toolset to manage those data, starting all the way from de novo genome assembly to pairwise and multiple genome comparison to constructing a pangenome graph, and finally storing these data and making them accessible through a programmatic data interface and API. Now, the reason for developing these tools may not be obvious, because there are many tools already out there in the public domain. And for example, for genome assembly, we often use a pipeline that consists of multiple public tools such as Minimap and Miniasm. And when we looked a little bit deeper into those tools, we identified that actually some of them have a large computational bottleneck. So whereas the genome sequencing becomes cheaper and faster every year, we see that these tools, they do not necessarily scale with these developments. And so there is a larger and larger computational cost to genome sequencing. And most of this computational cost is actually found in sequence alignment. So, and that not only goes for genome assembly, but that also goes for genome comparison, both pairwise comparison and multiple comparison. And so to try and solve this bottleneck, to make sure that we can scale up to this new pangenome world, where we have tens or maybe even hundreds of complete genomes for a given species available, we started looking at how can we solve this. Now, going back to the 80s and 90s, we already know that these genomes that plant genomes are a bit different from human and animal genomes. So they are much more repetitive. And so, for example, if you have two genomes shown here on both axes, and they share a common element, you can draw a kind of line as an alignment, indicating that this element is found in both genomes. If this element is present twice in each genome, you will make 4 of those lines. You can make 4 alignments between these 2 different copies in each genome. Then for 3 copies, intuitively, you know that there will be 9 possible alignments. But then for many of those copies, there will be many squared possible alignments. And the goal in trying to align these two sequences or aligning these two genomes is finding, let's say, the best collinear path, so the most probable alignment that shows how these two genomes look like each other. And this becomes a massive computational bottleneck in case of these plant genomes that are very repetitive. So in order to solve that, we actually went back in time decades, because actually in the early 90s, when KeyGene was founded, we had a technology called AFOP. And AFOP is based on restriction digestion of its genome, and then a comparison of those restriction fragments. And basically, we can do the same now once we know the sequence. So again, we can digest the genome with restriction sites. We can add the selective nucleotides, as we would also do in AFOP. So we know these from the genome, and then we can kind of align these restriction maps based on the distance between the restriction sites. And this technology scales very well also to multiple genomes. So for starting again with this pairwise genome alignment, we can represent this as a consensus, so flattening down this alignment of 2 genomes, and then add a 3rd genome to it. And by following the actual phylogeny of these genomes, so how they are different, how they evolve differently over time, we can actually build the most reliable multiple genome alignment out of these data. Now, also, we of course need to store these data. And as I showed previously, this idea of progressive multiple sequence alignment can be drilled down to locally collinear blocks, so LCBs that are parts that are shared between the genomes, and they may reflect structural differences, and so those structural differences you can represent in a graph, where each node is then an LCB, a block that is locally collinear between all the genomes, and each edge shows how these blocks are arranged in the different genomes. And so we built an API that stores both these genome sequences and the LCB graph, as we call it, and that allows programmatic access to this pangenome. And that's nice, because then we can develop external applications that can query this genome. And you can think of very simple queries like find me all the SNPs, or find me all the haplotype blocks that are present within a set of genomes. But also we can use this for gene homology detection. And of course, we couple this API to our Cropedia visualization software in order to view the contents and interrogate the contents of the pangenome and to make them accessible for breeders. Now, as I showed also during the previous talk, we developed a tool called Progressive STL that allows for ultra-fast multiple genome alignment at population scale. So, as a small example, we took 3 rice genomes from the public domain and compared them between several tools, and our Progressive STL technology can align these 3 genomes within a minute, whereas other tools take an hour or several hours to do so. Now, we can also scale this to larger genomes. So taking a polyploid field crop genome from about 4 gigabases in size, taking 9 of these genomes, we can align them with our technology on a single computer within 1 hour, whereas all the other tools did not finish even over the course over a week. And it doesn't stop with these large genomes. We can also scale to population sizes. So we can even align more than 50 brassica genomes, so an entire germplasm of brassica, within a day. And this allows us to investigate these pangenomes in much greater detail than we could before. Now, it's not only speed that matters, of course, because it's nice if you're fast, but it also has to be accurate. And so we compared our Progressive STL technology to some other aligners. And mind you, we had to take a very small data set for this, because all the other tools, as you can see here, they don't scale very well. So taking a single rice chromosome from 3 accessions, we could align that in 15 or 16 seconds. Whereas other tools would take minutes or even hours to compare these 3 relatively short sequences. And out of this alignment, we get what we call LCB, so these locally collinear blocks, where 2 or 3 of the genomes align to each other. And the number of LCBs says something about the structure of this genome. So a small number of LCBs means that these genomes are aligned mostly collinear, whereas a large number of LCBs would indicate many structural differences. Now, we know that these 3 rice genomes, or these 3 rice chromosomes, I should say, are relatively collinear. So we expect a small number of LCBs, and we see that many of the tools produce 1 or 2,000 of these LCBs, with a large outlier cactus, which produces hundreds of thousands of LCBs. And as you can see, these LCBs from cactus, for example, are much smaller than other tools produce. And our progressive STL produces LCBs of about 30 kilobases. So this means regions of about 30 kilobases that we consider collinear. Now, of course, if you consider everything collinear, you will have large gaps in them due to the real insertions and deletions. And if you don't consider, if you consider every gap a breakpoint in this collinearity, you will have very small LCBs. So what we found with our progressive STL technology is that we can build relatively large LCBs with a relatively moderate number of gaps in a very fast time. And this represents very well how these genomes align to each other. Now, PanGenomes from different crops, of course, have different complexity. And what we have seen is by looking at very small genomes like Arabidopsis, taking about 20 of those genomes and aligning them to each other, we see that most of the genome is actually the same, what we call the core genome. So those are regions where all these 19 accessions align collinearly. Whereas if we go towards more variable genomes like rice, which also has many different accessions, as you saw previously during the previous session, aligning 3 of those genomes already shows that the core is much smaller, and you see this large piece popping up of unique, which is basically pieces of the genome that are not found in any of the other accessions. And these often contain very specific genes for those accessions. And going a step further, we can actually look at these 9 field crop genomes, this PanGenome out of those. And there you see that approximately only 2/3 of the genome is core, and that there is now a large section of genome that is called dispensable. And this means those are regions that are present in 2 or more of the genomes, but not in all 9 of them. And so this allows us to dig into these genomes and compare features between, but also amongst those genomes. Now, as a small showcase, I want to talk a little bit about a Fusarium PanGenome that we made recently. So Fusarium is a genus of plant pathogenic species. This example is about a Fusarium species on an ornamental flower. And we have sequenced 3 of those strains and compared them in a pairwise fashion, as you see on these images. And what you see here is that these genomes are highly collinear and also have a very high shared content. And actually, we also found in the PanGenome that about 90% of this genome is shared amongst these 3 strains, and about 5% is unique and 5% is dispensable, so shared only amongst 2 strains. And when looking at the gene content of these genes, we looked at the BUSCO genes, so a set of single copy orthologs that are universally conserved. And we found indeed that each of those assemblies contains 1 copy of more than 99% of those genes. And combining them into the PanGenome, we still see that the vast majority of these genes is still unique within this consensus representation of the PanGenome. So we get nice multiple alignments of each gene in the PanGenome. And actually, that's an advantage, because the PanGenome also enables us to perform homology detection in a way that we could not do with only the protein sequences. For example, take this gene here. What we see here is the 3 Fusarium strains that we sequence, a multiple alignment of this, produced by our progressive STL technology. And you see here at the top that there is a small SNP, or perhaps a sequencing error, in one of the sequences. So this region of the genome is fully conserved except for a single nucleotide. But what happens with the gene prediction is that 2 out of those 3 genes get an extra exon compared to the top one, or the top one misses an exon. Now, if we perform protein clustering on these genes do not end up in the same cluster. But clearly, you can see from the collinearity in the genome that this must be the same gene. So just having a SNP or a sequencing error does not disqualify this gene as being the true ortholog. And so this is what the PanGenome, this multiple sequence alignment, now enables us to look at in a much greater detail than we could before. And when looking at this Fusarium gene space, looking at these 3 strains, we see that about 20,000 of those genes are conserved between the 3 strains. And there's a few thousand that are actually found specifically only in one of each of those strains. So this is the unique part. And there's a small part of genes that are found in only 2 out of 3 strains. And now if we look at how those genes are distributed over the genome, we clearly see a picture that we already know from literature, namely that there are some really disposable chromosomes within Fusarium that contain genes that are unique to a single accession. And of course, those are very interesting for the breeder in this case, because those may harbor the genes that cause either the resistant or susceptible interaction. Now, a second showcase I would like to talk a little bit about is about these brassica genomes. So we sequenced more than 50 brassica genomes and put them together within one day into a pangenome that comprises all these 50 genomes. And so we took a public tool called MASH that allows us to measure let's say, the distance between these genomes based on minimizer sketches, and we built a neighbor joining tree from this. So in order to represent more or less the phylogeny of these 50 accessions. Now, if you look, if we compare this now to how the pangenome represents these genomes, you can see that the genomes that cluster quite closely together in this tree also have a large set of shared LCBs. And in this case, shared LCBs means that the same region of those genomes is aligned to each other. You see nice dark blue blocks forming around the diagonal, where closely related accessions based on the neighbor joining tree are also much more similar in the pangenome. And then at the bottom, you see a few outliers that are completely red. Now, I want to point out a few of those outliers. So first of all, there's one on top here, and that's actually a public reference genome. And that genome is not as complete as the ones, because it is a lot older, so it's not as complete as the ones we sequenced recently, and it's also much more fragmented. And that means that within the pangenome, it ends up less well within the LCBs. And we see that, so there it looks much different from the neighboring genomes, whereas in the phylogeny, of course, it looks very similar. But also here at the bottom, something strange is going on, because the matrix turns completely red. And actually what we found out is that these are different species. So these are not the same brassicas as the one in the rest of the tree. And then somewhere near the bottom, we actually have some interspecific hybrids. So a cross between these different species and the ones on top. And you see this nicely not only within the phylogeny, but also if you look into the distance matrix from the shared LCBs, you see these blocks occurring. And this shows us that we are able now to, from this pangenome, also identify the similarities and differences between these genomes. Now, we can also use this pangenome, as I mentioned previously, to identify gene orthologs and to identify the homology between genes, but also, of course, to extract the variation within those genes. And shown here is just a random example, but in order to give you a feel that really this technology now enables us to look at 50 copies of a gene at the same time without having to do an annotation of those 50 genomes. We can annotate one genome and find the equivalent genes within all those 50 genomes just from the multiple alignment, and also identify the SNPs and indels, but also structural variations within them. Now, the final point I would like to make is why it is so important that this technology is so fast. Because, of course, it doesn't necessarily help you if it doesn't give a better result. So looking at this pangenome of the 50 brassicas, what we did is, what I plotted here is on the x-axis the number of genomes that share a certain fraction of the genome, and on the y-axis the fraction that they share. And what I would expect is on the right-hand side a large fraction of the core genome, and on the left-hand side a fraction of the unique genome, and in the middle is then the dispensable genome that is shared by a subset of the lines. But we don't even see a peak at the right. And of course, a part of that can be explained by those 3 or 4 genomes that were from a completely different species that we saw in the tree. But we also knew that this was not the result that we wanted. And so being fast, being able to do this within a day, also allowed us to play with parameters and actually very much improve this pangenome, making a much larger core genome and a much decreased unique genome by simply tuning our alignment parameters and running this again. And if you can imagine that this would take days, weeks, months for other tools, yeah, it's practically impossible to play around with this and get the best possible result unless you have only one set of parameters that always produces the best result. And so with that, I think I've shown some examples of how we now can build these pangenomes, that we now have technology to do this very rapidly, and also allows us to play around with a lot of parameters to make the best pangenome out of it. I've shown that we have an API where we can ask simple queries like show the SNPs, give me the gene orthologs, etc. But we're also looking ahead, and so we want to be able to in the future compare between pangenomes, so compare between species maybe, but also align reads to our LCB graph in order to do variant detection based on short reads or long reads, and then do genotyping and imputation of large populations. But also simple tools like a sequence search, for example, to develop primers that can amplify a region within all 50 or all 100 genomes in the pangenome. And perhaps the thing that we learned from this gene orthology search is that we really need a consistent feature annotation. So a good annotation for genes in all the genomes and not based on just a single genome or all independent annotations. And so with that, I would like to give the word also to my colleague, René, who will talk more about the downstream applications of pangenomes for crop improvement.
Thank you very much, Erwin, for that very in-depth insight into what goes into the construction of the pangenome, the tools, not just that they impact the turnaround time, but also the quality. What I also like that you briefly touched upon is that how these improved efficiency can add value for the breeder in their breeding programs. And that nicely connects with our next speaker, and that's René Hofstede. René is the program scientist at KeyGene, and he's leading the vegetable and the fruit research here at KeyGene. Prior to KeyGene, he worked at a breeding company for 17 years as the head of the cell biology and the molecular genetics lead. Therefore, René likes to take a very pragmatic approach on technologies, as in how their application can be translated into plant breeding. So today, again, René will share with us his view on the application of pangenome for plant breeding. In the meanwhile, before I hand over the screen to René, if you have questions for Erwin, please do type that into the chat box. René?
Okay, thank you, Sheetal, for the nice introduction. But let me explain a bit more of what I do in my daily life. And that's my group is involved in applied genetic research. And that involves links like making genome assemblies, resequencing, SNP mining, all of that to get SNPs to do the genetic studies like linkage analysis, the BSAs and the QTLs. So pangenomes are very relevant for my work. And what I will do now, during this presentation, I'll try to go from the current applications to the future applications in only 15 minutes. So that's quite a big ground to cover. So I'll first show examples in somewhat more detail, and then jump to some other examples with showing less details, which I hope you will understand by then. So if you go to the example, that's a typical QTL analysis. So what do? You do your phenotyping and genotyping of a population and do mapping, ending up with a lot plot, which shows you your QTLs. Next step will be to find recombinants in your QTL region and genotype those again and try to position your phenotype in your recombinant map. And once we've done that, then we typically go to the Cropedia and go look for candidate genes in the region which could explain the phenotype that was mapped there. And lastly, of course, we then enter into the gene validation to actually prove that the gene is causal. So when I explain that I want to have pangenomes, I often get the question, yeah, but why? Eh? Because what you show, you can also do with SNPs. So what's the added of pangenomes? To explain that, I need to take you back to the recombinant map and show you the actual mapping of all markers on the genome. So you see the light blue lines in which the genetic position of a marker connects to a physical position on the ruler of the Cropedia. But as you can see in the recombination bin, so the block on the figure in the middle, there are no recombinations in between, and you see lines crossing each other when you physically position those markers, which makes sense because you're not sure about the genetic position. But there's one marker going from this block to the right side of my ruler, so outside the region I'm looking for. In the time when we didn't have whole genomes and pangenomes, we would argue that probably an artifact of genetic mapping. So maybe a wrong phenotype, maybe some sampling errors or a wrong genotyping. But now we have more and more genome sequences. We think of it differently. Because you can imagine a situation like this, that the P1 that shows you the genome of your reference genome. The light blue line again depicts the marker being located outside your recombination bin. But it might very well be that in the second parent, for instance, the resistant parents, there was a translocation by which this marker and its surrounding actually is positioned within the QTL region. That has a quite dramatic consequence, which is that all the genes that are in the reference sequence located in this region now all of a sudden become interesting candidate genes. So you have more genes to choose from, and well, in some cases those do contain the causal gene. Also, it allows you to look differently at the genetic distances, because it might very well be that the block in parent 2, the dark blue block, doesn't have a counterpart in P1, and therefore there is no recombination. Therefore, it actually fits exactly within the recombination bin, and the reference position is false, I would say. So how much evidence is there of differences between genomes? Well, for that, I show you some examples that we did a de novo assembly of 4 accessions of a certain crop. And we start looking for structural variants. And as you can see in this graph, for instance, accession D, the lower one, it has over 30,000 structural variants. So a dramatically high number, I would say. Then you can start looking at how unique are these. And that's what the second graph on the right explains, that about 60% of all indels and insertions are unique for a certain accession. Then the next question will be, okay, how does this structural variation translate into gene content? Because that's probably more important for your traits. Well, we had another example of where we sequenced 3 accessions, and we had to look at the size of the structural variants. And the second row from the top, it shows you the number of indels that have a size between 1 and 10 kb, so gene sizes. And as you can see, there are thousands of those between the lines. And you can also see that already from the gene annotations. So you see here 2 gene annotations, public annotations from Pepper, where in the top panel you see 4 genes that are not annotated in the lower panel, which is a different accession. Some of the differences are caused by actually making the track and positioning the genes on your ruler. But for some other cases, for sure, there's differences in gene content. So having said that, I think if I have PanGenomes, it can help me very much to elucidate our gene clusters, which are notoriously difficult to assemble in genomes. But also it will help me find genes which have causal structural variants. And there are more and more examples coming that a causal variant is not a SNP, but it's actually a structural variant. So from a distance, I would say PanGenomes, it will help me in my SNP mining. It will help elucidate or find structural variants. Thereby, I can create more accurate haplotypes. And that allows me to look at differences in gene content. And all of that, it will facilitate gene cloning projects. Well, then the next question I get from breeders often is, why do I need to clone my gene? Because a marker also works perfectly. Then I show you some reasons for that. I think the first 2, or first 3 actually, so intellectual property or generating novel alleles through our mutation platform, keypoint breeding, that's one very good reason, I think. But a reason that's often forgotten, that's the validation of markers for complex traits. And I have a graph showing that or explaining that. So typically you take a germplasm set, you screen a number of markers, and you find a picture like this. So a set of susceptible lines on the top, and a set of resistant lines on the bottom. And for instance, the green haplotype on the bottom, it explains your resistance in, well, let's say a third of your resistant lines. Well, in this example, there were more loci involved, and you can already see that these pictures, they become very much complex if you go to longer loci, because then your number of haplotypes increases dramatically, and if you add more loci. So pretty soon, let's say if you're looking at 4, 5, 6 loci, you cannot work with this anymore. And then this is not the way to prove that a marker predicts the trait right in your germplasm. I think that's where gene cloning comes in, and it can help you to allow you to use markers. Of course, I can also reverse that reasoning, that if I have technology for gene cloning, then I can start looking at more complex traits. ending up with markers for breeding. So that's the example in more detail. So now I'll jump to some more, some other examples. And the first one is about haplotyping. So I have here an example of 72 lattice lines that were genotyped with 1,400 SNPs. And if I look at a specific locus, in this case the linkage group 1 locus for the DM cluster in lattice, you notice that there are a number of H-scores and a number of novel haplotypes in two of the PI numbers. Those are shown on the right-hand side. Well, if I think I had a pangenome of all these accessions, I would on one hand can elucidate my H-scores, because they probably or possibly are the cause of gene duplications. But I could also more accurately look at the genes underlying my haplotypes. And thereby I could distinguish the haplotypes in the two accessions on the right, or whether they are true alleles of genes, or that they are caused by a recombination between one allele and the other. So they do have the same gene content. The other example of haplotyping is if you add pedigree data. So here I show an example of the pedigree of the Latero lines, which are well known in the tomato community. So I think it would be great if I had the genome assembly of all 32 or 33 accessions, that I could actually follow my genes through the pedigrees to see which genes cause which resistance. Well, it becomes even more interesting if I expand the numbers. So here you have the pedigree network of about 600 public tomato lines. I think it would be great if I could follow gene content through this pedigree instead of haplotype content, because then it's far easier to make a link to the actual traits. An example from another side is recombination. Look at recombination prediction, I would say. So as we all know, recombination that's based on chromosome pairing in meiosis, that pairing and crossing over is influenced by genomic features. And as was shown by Sefke and Demirci a few years ago, that sequence and shape of the DNA can predict your meiotic crossovers. That means if I have multiple genomes, my prediction of recombination improves dramatically. And so that leads to the situation that if I would have pangenomes, I'm far better equipped to predict the results in breeding and developing research populations. But also to take a step back to the beginning to explain situations where markers are not located where you expect them. The next example is going to complex traits. So currently, we're using GWAS experiments on germplasm sets. And by doing that, we can detect SNPs that are specific for breeding programs. And I show an example. The panel on the left, it's accessions from a breeding program selected on high sugar. On the right is a breeding program not selected for high sugar. And by looking at the allele frequencies, you can detect SNPs that were selected for. And one of the benefits, as you probably will realize, is that you do not need very accurate phenotypes, as long as you know it has been selected for and the locus can be selected for. So then going one step beyond this, so if I can combine this with having pangenomes that cover all genetic variation there is in a certain species, and combine it with shallow sequencing that will allow me to infer the alleles present in each of my accessions, I'm far better equipped to look at complex traits and start elucidating those. And from my side, I think there are two outlets of once you have detected those loci, and one is towards breeding, which is the variety designer, and the other one is the allele factory. And I'll explain both. So I have to go one step back. Yeah. So the variety designer, it boils down to once you have selected your top 100 loci and selected your preferred haplotypes, then you can start using software like the Prospect breeding software we have developed to design your breeding strategy to combine those loci and alleles in the fastest possible way. And it can be based on classical breeding, prediction of recombination, but also creation of new alleles like mutation breeding or gene editing. And once you have designed your strategy, of course, then you have to carry it out. Then the outlet towards research. I think when we start doing research on complex traits, we will be faced with multiple genes that are interacting. So we need a high throughput generation and validation of alleles. And I think that can be based on gene editing in a magic-like approach. So in the picture on the right, you see transgenic line A having the Cas9 and two other transgenics, B and C, having guide RNAs to target 10 different genes each. But then you can start combining them, selving, and making basically a population that segregates for different combinations of novel alleles for all of those 20 genes, which will be a great tool to study the effects. And if needed, if you need more genes, you can add a parallel magic to it. So in summary, I think PanGenomes, it will give me a higher resolution and other type of data, which will increase the insight in gene clusters, will facilitate gene cloning for complex traits and prediction of recombination. Pretty soon, I guess, that will be combined with population genetics to identify the loci for more complex traits. And as a last step, I think once we have that, then we'll move on to the use of variety designer on a larger number of loci and start making actual allele factories. And with that, I'd like to thank you.
Thank you, René. Thank you very much for this interesting talk again. You not just explain how the pangenome can be used or applied in plant breeding, but you specifically, what I noticed was how it is of a high added value when you are dealing with complex traits especially. Just opens up a new avenue to information, and that can be translated into applications. So if you are excited or have questions for René, please do type that in the chat box. With that, comes to introducing to our final speaker of this hour, and that is Professor Ellen Session. Ellen is the Senior Principal Scientist and the Crop Genetist Team Lead at BASF Crop Science. He will be discussing today with you his work in cotton and soybean crop. And he will tell us why he's longing for being able to use pangenomes instead of the linear reference genome. Alan?
Hello. Thank you for that introduction, Sheetal. And thank you, KeyGene, for organizing this seminar and inviting me to share what we're thinking about where I'm working. My name's Alan. I work in BASF in a site in North Carolina, United States. We work primarily with breeders, cotton and soybean breeders, and we provide genotyping support for them to help them make selections both for simple traits and complex traits. So genotyping is my whole world, and so I will tell you today how I think we'll be able to improve on our genotyping for what we're working on primarily, and that is predictive breeding. So in my talk today, I'll just start with predictive breeding and how we're using sequence-based genotyping and how that is somewhat limited from an information content. I'll move into a few pangenome data structures, present a few examples from the literature, and close with some final thoughts. So the primary task we have in our lab is genotyping for predictive breeding. We work on cotton and soybean, and I show you, let me see if I can get a pointer here. I show you a generalized breeding scheme for inbred crops like soybean or cotton, starting with founders and parents and creating populations that over successive generations are trialed in the field to select lines with optimal performance. So this is the traditional way, of course, and we're moving to exploit predictive breeding to make this more efficient. Predictive breeding is based on a prediction model that is made made of genotype data and phenotype data from our founders and our parents. high-density genotype data and multi-season field data that can be integrated into a prediction model whereby you can genotype progeny, plug them into the prediction model, and get an estimate on the performance in contrast to trialing those lines in the field or in addition. The key to genomic selection or predictive breeding is this is the accuracy of your model, and that's based on your genotypes and your phenotypes. For the genotyping component, we're doing mostly short read sequencing for our genotyping, either whole genome or genotyping by sequencing. And we are interpreting those sequences against a linear reference like most people. So our linear references are beautiful. They guide our interpretation of new sequences through alignment, like this one shown here. But they do have some limitations, and one is it's essentially based on the reference represents a single haplotype found in that species, and that is not indicative of the entire species, and it also causes some bias in the read mapping to that reference. So we typically see biases in calling alleles. We have reads that map the reference allele more favorably than variants. So we're biased in calling reference alleles. We're also biased in not being able to call genotypes on sequences in our reads that are not present in the reference. So that limits our ability to call small indels in all structural variations. We can call SNPs quite well, and that can suffice, but we'd like to look at the structural variations as well. So the last part of that limitation in the last slide, where we have reads that contain sequences not present in the reference genome, has become all too apparent in the last 5 to 10 years as we sequence more individuals and compare them to our reference genomes. So as Erwin mentioned, now we have concepts such as core and dispensable genome, because what we see when we sequence in any species a number of individuals is that there's a large fraction of genomic DNA which is segregating across individuals as presence or absence. And so when we align individuals, we commonly see these core and dispensable genome fractions as segments, with the core genome being conserved across lines, whereas the variable fraction is either unique or absent or present. This table shows a summary from recent studies across a number of species. This is really a universal aspect of biology. The variable fraction in different species can vary depending on the study, how they did the study, but again, it's typically quite significant. The most exciting thing for us in predictive breeding about this variable fraction is the content. So I know the content, of course, is enriched for repetitive sequences, but it also contains genic sequences. And all of these studies and others have shown that the variable fraction of the genome is enriched for proteins that encode biotic and abiotic stress responses. So enriched for environmental response genes. So PanGenome, that's been introduced nicely by Erwin. And the best way I find to describe the difference between or what a PanGenome is by contrasting it with a linear reference model. So in a linear reference model, we're interrogating our sequences individually against the reference. So everything is compared to the reference. In contrast, in a PanGenome model, we're really comparing every individual to each other. And so we're able to better represent the core and variable genome spaces better because we're not suffering this reference bias by comparing everything to one reference. And we're also able to make it more efficient by combining sequence variation into a single data structure. So when we see pangenomes in the literature, they're typically of two general types, unfolded pangenomes and graph-based pangenomes. We see a lot of studies like those I listed on the previous slides where there is a wealth of sequencing done on a nice set of germplasm, and the differences are cataloged but not connected into a coherent data structure that allows exploiting the orthology relationships between those genomes. They're typically not enabled for genotyping with short reads, and they lack the compression for efficiency. Graph-based pangenomes, as introduced earlier, are really a simplification of an alignment, a multi-sequence alignment, whereby we can compress the information into a series of nodes containing sequences and edges, which are connections between those sequences. We can describe variant positions in the genome as a branch point in the graph, and we can also characterize individual genotypes as paths through that graph. So a series of nodes and edges describes the genotype of the individual. So graphs are very elegant. They include information about alignments of all the sequences within it. They compress the information, essentially that redundant information is compressed to make it more efficient. It's easy to represent individual sequences in that graph structure. And most importantly for us, it allows the read alignment to occur within a context of sequence variation. So how might we employ a pangenome in our predictive breeding programs for cotton and soy? I bring back my traditional breeding process, and I replace my linear reference in the previous slide with a diagram for a pangenome. We think having the pangenome be the central core of genotyping will lead to a number of improvements. One, genotype and variant discrimination and calling will be improved across structural variants, short indels, and large structural variants. So as mentioned, we're primarily focused on SNPs now because they're easy to call and they're easy to call accurately. Prediction models based on a pangenome genotype will also incorporate all of this structural variation. So it'll bring in all genetic variation as opposed to simply the single nucleotide variation. If we can have our variable fraction in our reference, we can also directly genotype that. And for predictive breeding, this becomes important if we are after those G by E genes that seem to be present in that variable fraction. And lastly, we can use the pangenome to optimize our marker panels for this progeny genotyping to minimize the number of markers we might need and make the genotyping more cost efficient. There are quite a number of tools published in the literature and also available from companies like KeyGene. I list three here. We have on this slide practical haplotype graph, graph typer, and variant graph toolkit. And these complicated diagrams, noisy diagrams, which attempt to show some differences between how these tools structure the genetic variation. So starting with practical haplotype graph, This is a tool that's being developed out of Cornell University and Ed Buckler's group. It's been designed for predictive breeding, an emphasis on the word practical, and the title of the tool refers primarily to simplification of the complexity. They simplify the complexity by only focusing on segments in the genome, which they term reference regions, as opposed to the entire genome. They also populate variation as consensus variation, as opposed to individual variation. So they collapse the complexity quite a bit to make things more efficient and try to not compromise information content in the process. So they just more specifically, they partition the reference, single linear reference, into ranges. For example, the gene space. You can use 20% of the genome. They add parent genotypes the traditional way with short reads. They call consensus haplotypes and link those through the graph with parent information, calling parental paths. And then you can genotype progeny against this graph with short reads, and the database will impute the best possible path through the database. In contrast to this plant-developed tool, GraphTyper and VG come out of the human genome community. GraphTyper makes very simple graphs, and VG makes more complicated graphs. GraphTyper will make what's called a local graph from simply a linear reference and either reads or variant calls in standard formats. So what I mean by local graph is that it only explores variation in local regions as opposed to across chromosomes or across larger spaces. The graph is, there are different graph types. One used by GraphTyper is called a directed acyclic graph. The only thing I want you to remember from that is that the edges are directed. So these arrows connecting nodes all point in the 5' to 3' direction. And so it can capture variation, but only variation that doesn't cause switching of strands. So it will not be able to characterize and harbor inversions and translocations. But it is a very efficient way to create a simple graph and to genotype the short indel space. VG Toolkit is the most complex tool listed here. It is under active development by the human genome community. The main difference between GraphTyper and VG is that the data structures in VG are based on bidirectional graphs. So it can harbor variations such as translocation and inversions where strands are switched. It can also be composed from all types of data, full assemblies, as in addition to short variant calls. So let me quickly summarize those three tools just to highlight some of the differing aspects. They all do retain some part of a reference bias. So for practical haplotype graph and GraphTyper, they are essentially based on a linear reference, but they incorporate variation into that context. VG Toolkit, in contrast, can make graphs independent of a linear reference. The data input to create a graph for all of these tools is also different. For PhD and GraphTyper, the requirement is less than for VG. The complicated data structure accepts more complicated data for VG. Each of these tools has a different graph structure. They all put genotypes into a VCF file for downstream application, and then each has pros and cons. Whether for PhD, it's minimal data to make, and it's currently being used for tools. The con is that it may oversimplify the information and compromise resolution. Graph Typer is easy to use. It does not call all variation, and it has a very limited user base. VG, in contrast, does contain all variation, but the compute requirements appear to be quite intense. It does have a proof of concept in soy, which is a very interesting read. So each of these tools has benefits and drawbacks, and I think we'll be getting, we'll also be seeing an addition to these tools with many more over the next few months in the literature. So to quickly summarize, variation-aware genotyping is needed to improve our genotype interpretation, and we're focused on that for predictive breeding. Improved genotyping of structural variants and the variable genome fraction will improve our prediction models and our predictions. Several approaches are being developed to enable PanGenomes, and there's not one unifying method at the moment. Pangenome construction for predictive breeding needs to have a different set of parameters than for gene discovery, for gene editing, for biotechnology, principally because of the cost pressure to make the genotyping very low. Companies like KeyGene, some of the academic labs I mentioned, and the human genome community as well, are very actively developing these tools, so stay tuned. There will be a lot of cool stuff coming out. So with that, I will stop and entertain questions if there are any. Thank you.
Thank you very much, Alan, for this very educative talk. What I really liked is that you also took time to really present the tools that are present in the public and the proprietary space. So depending on the research need, one can choose which to use or to start with. So I hope that was also very valuable for you all who are watching this presentation. So thank you again, Ellen, for that. With that, this leaves us for the question and answer session. So the panel is open to receive the questions. So I would ask my colleague Eric, can you present some questions for us?
Yes, of course, Sheetal, thank you. Let's start with a few questions for Ellen Sessions. And there is a very curious question from somebody called Taco. And Taco is very curious what NGS technology for de novo assemblies would be favorite now at BASF?
Well, I think the community is in all species is moving to long reads, both for, the phasing of the variation, but also the ease with assembly. So I think we're joining many groups moving in that direction. But again, we all have a lot of short read data as well. So we are challenged to exploit that.
Thank you, Ellen, for that answer. Eric, we can take one more question if we have.
Okay, thank you. There's a question from Chris, and he is curious about the predictive value of the models and the accuracy of the models. What do you think, Alan, is the accuracy of the best prediction model that you use?
I think it's going to depend. In our application, and I think in most applications, the goal is not to select the top 10 out of 10,000, but really to eliminate the bottom half of 10,000 and to focus on the good things. So the accuracy doesn't have to be completely accurate. The correlations to actual phenotypes will range, but we find correlations of 0.5 or 0.6 are sufficient to make effective genetic gains. And you can get better correlations with improved models. It all depends on the phenotype and genotype data being put into it.
Thank you. Would one of you want to add to that? Erwin? Okay. Thank you, Ellen, for that answer as well. We can take a few more questions. Eric?
Yeah, let's go ahead with the question for René. Say when you use a pangenome strategy for gene mining, why would it not be very important to have an accurate phenotype?
That's because if you start comparing two sets of germplasm, it's sufficient to know that a certain set has been selected for, because that will lead to allele frequency differences that you can detect. So you don't need to have the phenotype of each individual, so you simply need to know they're from a certain breeding program.
Mm-hmm. All right. I hope that satisfies the question asked. Eric, we have time for a few more questions.
Okay, let's move on to a question for Erwin, and it's both from Juan and Michiel. They wonder, is it uncommon, Erwin, to miss orthologous genes just from a single missing exon when you're using, for instance, software which is dedicated to find homology or orthology, unless you are very strict parameters, perhaps?
Yeah, so it depends, of course, on how much these genes differ. I mean, the criteria that are normally used are based on both length and on sequence similarity. In the example I showed, of course, the similarity was 100% because the only thing that changed was that an exon was skipped. But the length, yeah, if this takes off 20% of the length, for example, then you may fall below a criterion of 90% length conservation, and you don't find it anymore. And specifically with these more noisy sequencing technologies, the quality of the final consensus is not always 100% yet. So of course we're moving in that direction, but before we are there, I think these tools can help us a lot by looking at genes from a pangenome view rather than looking from the individual annotations.
Right. Yeah. Thank you, Erwin. We have time for one more question. Erin?
Okay, thank you. There's another question for Ellen. Ellen, you nicely showed us 3 of those tools to make graphs to show the pangenomes, and including drawbacks and benefits of those 3 tools. But in practice, how difficult do you think it is to get from an unfolded linear presentation to a graph presentation?
Well, to make very simple graphs, it is quite easy for most people with graph typer. The complexity of that graph and the utility might not be as great as others. And then there's also the practical haplotype graph, which allows really minimal data input to create these. I think a tool like VG, that is quite complicated and more difficult to use. So I think it depends, and of course, the easiest way to go is to work with a provider like KeyGene or other companies that will help you along that way.
Thank you, Ellen. Let's take one more question if we have. Eric.
Yeah, there's one I would like to read out. I'm not sure whether I properly understand the question, Sheetal, and I'm also not sure for who the question is. Mm-hmm. Why is the VG got the limitation in plants? Because of the repeat sequence or others, just due to users or some genome sequence limitation?
I can take that. I think it's primarily the repeat structure. The graphs are too complex and too computationally onerous to traverse. So the example that's been published in Cell and Soybean, they did a very smart process of selecting the variants that the structural variants they populate into their graph, they remove all of the repetitive sequences. So in their case, they were able to make a graph that was quite effective, but I think it's the complexity quickly becomes overwhelming with VG.
Right. Thank you, Ellen. Eric, we can take one more question.
Yeah, and
have
it's a
one?
question that one could expect. It's about money. And since the genomics business is now moving from short read to long read, what are the implications on speed and cost of the PanGenome projects, for instance, at KeyGene? And perhaps René can say something about it. Of course, I know it's a complex question, but still.
And I'm sorry to say, but I don't know much about the costs involved.
So what we see is that the cost of genome sequencing is, of course, always going down with these new technologies being developed. And it becomes within reach now to generate multiple reference level genome sequences from those long read sequences. And yeah, I think this is also the direction we will be moving towards. What we see now is that both for ONT, but also PacBio, the two currently available long read platforms, the individual read quality is going up a lot. And this is getting close towards the short read quality, which means that you can actually also start using those long reads to do variant detection, both simple sequence variants, but also larger sequence variants, like the indels and SVs that you cannot properly detect with these short reads. So I think the field will be moving towards this direction more and more, where a lot of these applications that are currently being solved by short read sequences will also be solved by long read sequences in the near future.
Right. Thanks, Erwin. And if there is any specific question or request to know, I think you can always send us an email and we can connect with you later on. Well, actually, that brings almost towards the end of this webinar, but I have one question for you, René. You argued that the gene cloning will help us tremendously, and or if it's needed for the validation of markers involved in these complex traits. Is that the only possibility, or there are more options?
I think that depends on the number of genes. So if you go to really large numbers, I think you move into the genomic selection that Ellen also showed, and the modeling to actually validate the genes in breeding. If you go to, you could say, I do the same for a smaller number of genes, let's say 5 or 10 or 20 loci, and not do genomic selection or prediction modeling, my whole genome, but only on those loci. That's a valid method, I think. But in the end, or soon, that will be, I guess, more expensive than doing actual gene cloning.
Right, yeah. Thank you. Thank you very much. Well, that brings us to the end of this webinar. So thank you very much, all of you, again, to take part in this two-part webinar series of PanGenome. And I hope you all enjoyed it. Perhaps you still have questions regarding the topic of today. You can always send us an email, the one that is now will be flashed on your screen, or go visit our website to find out more details about these topics. And if you have questions for our speakers, please do send us an email about that as well. Shortly after this webinar, you will receive a review email from us. It would help us a lot if you would send your input. It helps us to improve our webinar even further. And before we leave, I'd like to take this opportunity to introduce to you to our next event. It's a flagship event that is going to happen in April this year. That's Crop Innovation and Business. This year, it will go virtual. It will be held on 2 days, 12th and 13th of April. And in these 2 days, in the first day, we will be focusing on innovative technologies for crop improvement. And on the second day, we'll be focusing specifically on innovations in vegetable breeding. So if you're interested, please do sign up for this event. We will be sharing the link here on the screen as well. And you're most welcome to sign up for this event as well. And again, I would like to thank you all. Be with us And especially I'd like to thank our speakers, Renee, Erwin, and special thanks to you, Ellen, for joining with us and for sharing your view on PanGenomes and your research work, and also giving us more idea about how widely the community that is interested in this can make use of this beautiful tool. So thank you very much, and I hope to see you all in April. And until then, goodbye, stay safe, bye-bye.